It was originally 3.3, but this is the last module of today's workshop. Okay. About secretion and absorption. So here's an interesting picture of a simulation where one cell is secreting and the other cell is absorbing. Don't forget that workshop is live streamed, recorded, and distributed. And if you didn't notice yet, my name is Pedro Castel from Brazil. The topics of today is secretion, uptake, and secretion and uptake, okay? So secretion and uptake, to be short. Let's start with secretion and exocytosis. So how secretion happens in biology. So the cell produces stuff inside in the Golgi apparatus or in the cytoplasm, and in the Golgi apparatus, it can be bundled inside vesicles and transported to the membrane. Once it reaches the membranes, the vesicles can attach to the membrane, or it can dock in a secretory protein called porosome. So for people not familiarized with this biological process like Julio, so what's happening, imagine the cell is this, so this is the cell, and my head is the Golgi apparatus. So I produce something and a vesicle forms with that, for example, enzyme inside the vesicle, and the vesicle travels to the membrane. It could be through the microtubules. So there's a microtubule here, and the vesicle crawls over the microtubule. Once it reaches the membrane, it attaches to the membrane, and it does like this, attaches to the membrane. And when it attaches to the membrane, it releases the chemicals into the outer space. Or it can dock into these secretory proteins, which are transmembrane proteins, very complex process. It docks into this, instead of merging to the membrane, it docks into a secretory protein, a porosome, and the porosome releases all the substances inside the vesicle, and the vesicle keeps existing. So it doesn't destroy the vesicles, okay? Either way, both porosome and merging vesicles either are modeled by secretion in our point of view, okay? So you can model inside the cell, the specifics of the cell using compartmental, using compartments in CompuCell. You can actually model the vesicles. It's complex. You can do it in CompuCell. In this way, you would distinguish both kind of exocytosis. But for our purpose, they are both modeled using secretion. When you are modeling secretion, there are three important aspects you want to ask. For example, where the secretion occurs, whether secretion happens in contact with something. So for example, when a cell is, for example, when a macrophage is in contact with a certain type of tissue, it may release a certain type of chemical, okay? And you can also ask you if this secretion is, if the cell secretes the chemicals at a constant rate, or if it holds the field at a constant value. Let's start with the first one, where secretion occurs in respect to the cell, okay? It can happen, for example, in all cell pixels. So the entire cell secretes chemicals. It can happen inside the cell, in the center of mass. It can happen only in the boundary of the cell. It can happen in the boundary inside or outside. So if you draw your cell here, secretion can happen in the boundary, but in the inside interface or in the outside interface, right? So these are all options you can actually implement in CompuCell if you want to increase the complexity of your simulation. Otherwise, you just go with all pixels or center of mass, depends on the situation. Secretion in contact. So for example, cells inside a cluster, let's say they don't secrete anything, but cells outside the cluster, they secrete something. So in this case, you can implement the secretion in contact. In this case, cells secrete something in contact with medium. Okay, I will demonstrate that to you. And you also can implement a constant flux mode versus a constant value mode of secretion. And the differences are pretty obvious. For example, if you hold the field at a constant value, if you plot the graph of the chemical concentration in the X component, for example, you will see that it grows. Inside the cell, it's a constant concentration of 10, for example, in this case, and it decays in the other side. On the other hand, if you model it as a constant rate of secretion, it will keep secreting. So you will see this kind of behavior where your chemical concentration spikes instead of holding a constant concentration value. You can import your concentration in your simulation in two ways. The first one is from the wizard, where you just select the secretion option in cell properties and behaviors, and it will appear in your XML file. Or you can go to your XML file in case you still don't have secretion, and import from the to edit menu. So in the XML, once you import your secretion, you can set it up. So for example, here are the options. Cell A secretes 0.1, its secretion on contact, constant rate, and it secretes also on contact with medium. So this entire line means that cell A secretes at a constant rate of 0.1 if and only if it has contact with medium, and contact is shared interface. The line below is constant concentration being held by the cell at the value of 1. And you can also set a constant concentration being held by the cell only if it is on contact with medium. So all combinations of modes of secretion you can implement in your model. You can also secrete in the steppables instead of the XML. Actually, everything you do in the XML, you can do in Python. So in Python, there are actually more options for you. So for example, secrete inside cell at the boundary with something, secrete inside cell at the center of mass, secrete inside cell with constant concentration, secrete outside the cell at the boundary with something, secrete outside, and so on. Okay, there are many options for you. So the main thing you need to be careful is that you must import a secretor object. So this get field secretor, this object is the secretor, and it must carry the name here, oxygen. So you give the name of the field you want to secrete, pass this object into a variable, a placeholder, which in this case is secretor1, for example, and you can use it whatever, however, the way you want, okay? There are extra options as well. So for example, you can calculate the total field integral. So in all space, the amount of field you have using this total field integral. Yes, module. Yes, this is it module. Yes. So the secretors carry this module called total field integral, which gives you the integral of the field across space. And it also, you can also use this method, not module, I'm sorry, method. It also carries this method amount seen by cell. So the total field inside the cell, okay, may be useful for you. So for example, if you are building a cancer simulation where the cells, in order to make the cells proliferate, you want to know what is the concentration of oxygen, because below this concentration, cells cannot proliferate. So you may use this secretor dot amount seen by cell. You can acquire the concentration of the field inside the cell. And there is this other, let's say, syntax, this secret inside cell total count. If you add this total count to the method, it will track the amount of field secreted by the cell. So you just specify dot underscore amount, and you pass it into the tot variable, and you can just print it. Just if you print this tot, it will give you a number. So the amount of field the cell secreted so far, since it's alive. Okay. So now I will demonstrate for you two things. The first one is implementing the secretion in CC3D. And the other thing is building a histogram. So how is this demonstration? So I start with a pack of cells in a round cluster. What kind of object is the secretion? And amount of field like a source? The second one I didn't understand, but the secretion is, I don't know the type of object, but it's probably a class with methods and properties. So for example, the methods I was showing to you, these methods are functions inside the class. So starting with a bunch of cells, I set the cells to secrete only contact with medium, and as they separate, so I set a high contact energy between cells. Okay, so they separate, and as they acquire more contact with medium, the cells inside start secreting this field. And if we plot a histogram of how much the cell is secreting per Monte Carlo step, as time goes on, we see this histogram changing, right? So you can get the simulation ready from the folder, the student materials 3.3, I think, folder. You can get the simulation ready to go, but I will build the, yes. So yeah, James is correcting me. I used the wrong terminology. So the integral of the field would be better called material in the field rather than amount of field, okay? Because the field is concentration. Okay, so first step, create a packed group of cells that repel each other. I'll open my to edit. So this is not a guided exercise. This is a demonstration. Let me first. Yeah, so the project panel is right here. New project demo. I don't know, Pedro. Okay, whatever. Cell A, why not? And here you have to set a chemical field. So let's set, for example, chemokine, nutrient. So we want the cells to have some shape, right? And we want them to repel each other. So contact and volume. We also want them to secrete. So we also have to import the secretion plugin, okay? Next, okay, so in this wizard, we want cells to have a uniform secretion or constant concentration. Constant concentration. No, uniform. Doesn't matter. And the secretion rate of, for example, 0.1. So here it means that the field nutr is being secreted at a rate of 0.1 in the cell medium. That's not what we want. We want in cell A. And we can already set on contact. So, for example, cell A secretes 0.1 at the rate 0.1 on contact with medium. So we select medium, add on contact. So in contact with medium, add entry, and there we go. Okay. So you can set secretion from the wizard, but that's not what I am going to do. I will set it on the Python because it's more complete. So secreting from an epithelial cell, say virions, will be implemented using secrete outside cell at boundary for a 2D simulation. I don't even know if it makes a huge difference, but I would probably secrete in all cell pixels, but I'm not sure if it matters actually. Okay. Okay, so this is my Python. In the XML, I will comment everything. I will just leave the secretion loaded. So there we go, it's loaded, nothing inside. We'll set it on Python, so we don't need it. We don't need any specification inside here. And for the diffusion field nutr, I will remove the secretion here because it will mask my simulation. Okay. I will fix these boundary conditions, set them to periodic by default. Yes, it secretes, Chile. It gets overwritten by the secretion plugin. So by default, I recommend you just remove this part if you are modeling secretion in the secretion plugin rather than in the diffusion steppables. Okay. I remove the diffusion, I remove the decay. I don't need these for now. Yeah. So probably just a global diffusion constant in the medium, just so we control better the total amount of field. That's it. Okay. Yeah, I don't need these lines. The simulation is very simple. Periodic everywhere. This is our packed cells. Let's run to see what happens. These are our cells. Okay, perfect. If I come and see the field, I should see nothing, which is correct. So 0 everywhere. I have no field. And here in the steppables, I will start secreting. So I will take advantage of this loop over cells. So I will remind you, this loop for cell in cell list loops over all cells in the simulation. Okay. And here I can import from CC3D Python, secretion uptake. I can select secrete at boundary on contact with, or simply, yes, that's it. Secrete inside cell at boundary on contact with. Could be outside, but it doesn't matter. Okay, so secrete inside boundary on contact with. Yeah, there we go. Here we get a bunch of syntax. For example, this is the acquisition of the secretor object. We don't need to acquire the secretor object every time we loop over a cell. We can acquire the secretor just once outside the loop. So let's move it out. This is a little bit more optimized, okay? In the field name, neutral, I'm sorry, I would type oxygen. So this, the name of the field must be the same as the name here, okay? All right. And the rest, I will delete these lines, the comments. We have two useful lines here. So the first one, secrete inside cell at boundary on contact with this will apply secretion. And the line at the line down here, it does the same thing, except that it keeps track, sorry, it keeps track of the total amount secreted by the cell. Each Monte Carlo step, it's not throughout the simulation, okay? It's each Monte Carlo step, the amount of material secreted by the cell, okay? So as we want to build a histogram, we must track the amount secreted by the cell. Okay, so let's get back to my slide. So I want to plot a histogram of how much the cell secretes each Monte Carlo step. Okay, how many cells secretes that quantity of field, each Monte Carlo step. So I must keep track of the amount of field secreted. To do this, I will use the line on the bottom. Okay, so instead of the line, this standing secretion here, I will use the total amount, secretion inside cell at boundary on contact with total count. Okay, so the syntax is ready to go. Dot amount, I'm tracking the amount secreted, and this value comes to this variable dot amount, right? These are the parameters. So cell is our secreting object. So if we are looping over cell in all cell list, each cell of this list will secrete, okay? So if instead of this implementation, we want, for example, a cell of a specific type to secrete, we will do cell list by type and self dot cell A, for example. So now you would be filtering by type, but in our case, all cells in the list, okay? So all the cells in the list are secreting 300 material per Monte Carlo step. This is not what I want. What I want is 0.1 per Monte Carlo step. And now in this list here, the third argument inside this function is a list of the type of cells which your cell must be in contact with in order to secrete. In our case, the cell type is medium. So just type medium in the list, and your cells will only secrete the field if they are in contact with medium. So now I run the simulation and see what we got. All right, so only those cells on contact with medium are secreting, and all the cells in the middle, they are not doing anything, only POTS stuff, right? So now we want the cells to separate. So as I designed the exercise, I want the cells to separate so it's more interesting. And as they separate, they start secreting more and more. In order to do this, I just change cell A to 30. 30. Now, yes, there we go. Now they start separating. And as they separate, the cells inside the cluster start secreting. All right. So now let's plot a histogram. So how many cells are secreting a given amount of field? So in order to build this histogram, it's very simple. We define a histogram in our start function. You come here, Python, scientific histograms, add histogram plot. So it even tells you that it must be in the start function. Click here, initialize the histogram. So histogram of secretion in the y-axis secreted material. I'm sorry, the y-axis is the number of cells. The x-axis is the secreted material. We want just one histogram. I will call it sec because it makes sense. That's okay. 200, more opaque. Now we want to plot this histogram. So let me delete this useless line. Python, scientific histograms, step, add histogram. Okay. Bunch of stuff. I only need one. It's only one histogram. The histogram sec. Okay. It even gives me the variable here. And I must set the input of an array to calculate the height of the bean. I will put 5 beans because I tested it already. And this array will be the array of the total amount secreted by the cells. So I will call it, for example, secreted material list. Okay. So for each cell, for all cells in the simulation, I must put all the total amount information I have into a list and import this list into the histogram method. Okay. So I will define this list. So sec mat list outside, empty list. I must define an empty list always, otherwise I will be appending to the list which already exists, already is filled. So doesn't make sense, right? I will be reusing old values of amount secreted. Doesn't make sense. So you always reset your list, and inside this for, you can append to this dot append. You import the amount secreted by the cell amount here. All right. It doesn't look difficult, right? So now let's see what I got. Oh yeah. A very powerful tip here, window tile. So I don't need to adjust everything. It automatically adjusts for me. So in the beginning of the simulation, I have 400 cells secreting 0. So this is 0 amount secreted. So I have 400 cells secreting 0 stuff, and only a handful of cells secreting something. Okay. And as time goes on, this cluster will break apart and we'll see more cells secreting the field. So as time goes on, we see the time evolution of this histogram, which is somewhat interesting to watch. Okay. This simulation is, you can find it in the demo materials, ready to run. And the histogram is a bit crazy because we are not fixing the X and Y coordinates, but this is also a possibility.
Sorry, one thing you might want to show them is hovering over it and the control you can do with the right click over the plot.
If I just perturbs it, it stops shaking. I mean, not the histogram. The histogram will keep shaking. What stops shaking the X and Y coordinates. So this by itself is already a good feature. I can zoom in and zoom out. I can refit it into a better size. And if I left click, there's a bunch of options. So I can, for example, manually set the X-axis range or hit it auto. If I hit auto, it will start shaking again. So go back to craziness. If you set manual, it stops. Okay. Mouse mode, I don't know what it does. Plot options. I can plot logarithmic scale. So let's put, for example, logarithmic in Y. Let's see what happens.
Careful, some of those may crash your player. Not all of them are implemented.
Oh, dangerous. That's really dangerous. Okay.
The transform ones aren't implemented.
Okay. So at least log scale works, which is the most useful probably of all transforms here. Yeah. You can apply a derivative, I didn't know. You can average, I'm not sure if it's implemented correctly or not. The grid should work, right? Show grid. Yes. So you can remove the grids. You can change the opacity of the grid.
The transform ones don't work.
Okay. And points. Well, I think as it's not a point, it doesn't make anything. But one of the most useful ones is export. You can hit export. You can set options, for example, separate date by comma or by tab. Those are the two options. The precision of your data, so how many digits you will truncate, etc. And you can set, for example, you can export at a CSV. So, for example, CSV you can open in Excel, right? Am I wrong?
That will work. That probably will not work on NanoHub though, because it won't have write privileges, but on a desktop, it'll work. And saving the image file will also work.
Okay, so for example, let's put it in here. Temp, save. So now if I go to that folder, my temp, oh, it's already readable by Excel. So it basically exports as an Excel file. So here I have my histogram here. I could even plot, for example, insert. Yeah, there we go. But I have to warn you, if you use Excel to plot things, the police of scientific correctness will go after you. So if you want to do the exercise by your own, you can do it. The material is uploaded in the folder, but we will cover uptake now, right? And uptake, like exocytosis, is a complicated process. And in the case of endocytosis, I think I talked wrong. So endocytosis, like exocytosis, are complicated processes. Endocytosis is very interesting because the cell membrane has to engulf the outsider, the molecule, for example, or the bundle of something. It has to engulf it, or it could be a bacteria as well. So this engulfment is actin mediated. So the actin form those fibers that protrude the membrane outwards, and it involves the membrane involves the intruder, the bacteria, for example, and it cuts the membrane, and you have the intruder in a very nice vesicle. Okay, it could be bacteria, could be glucose, could be anything. Okay. So important things to consider when you're modeling uptake: the rate at which a cell absorbs something. So for example, a single cell will not absorb 1 kilogram of oxygen per second, okay? And the maximum absorption rate. So for example, some cells can uptake a given chemical up to a certain amount per second, and then it stabilizes. It cannot absorb more than it, okay? So endocytosis, you could model, for example, as a sink, but there's a problem with it. If you model as a sink, your your field outside could go to negative values. So once the field reaches zero, your cell will keep sinking, keep sinking, keep sinking, so your field goes to negative values. And if you only model as a decay, if you give the cell a ton of oxygen, it will absorb, for example, 100 kilograms of oxygen if you set 10% of decay, a 0.1 decay constant. So it doesn't make sense either. So for example, a sink makes sense if your field is very high. So for example, if you give 1 ton of oxygen for your cell will absorb a fixed amount of oxygen per second. If I give on the other hand, if I give a very low amount of, a very low concentration of oxygen, your cell will uptake a fraction of this oxygen, okay? Or it may as well uptake everything and stop taking up the rest of the oxygen because it's zero now if it takes everything. All right, so there is a combination of both sink and decay that we can implement to model uptake, which is for low values of field, we implement a decay, which is a gamma times C times the concentration, proportional to the concentration. And if the field is very high concentration, we can implement it as a sinking. So at which point does the cell uptake as a decay? And at which point it uptakes as a sink? So we define a value for it, a threshold concentration, which we call saturation concentration. This term makes sense because the cell absorbs more and more and more as you increase the concentration of the field, but it saturates at a certain point. So it reaches a plateau where it cannot absorb more, even though you are increasing the concentration of the field. All right. And in terms of implementing uptake in CC3D, the uptake already comes with the secretion plugin. And all settings are identical to secretion, except that you cannot secrete uptake in the XML. You can only set, configure your uptake in the Python code inside the steppables, not in the XML. Yes, Edgar. But the problem is if you implement as a sink inside the tumor where you have 0 glucose, you start getting negative glucose concentration. So instead of modeling as a sink, you model as an uptake, where for low concentration values, the field decays rather than sink. Because the decay is proportional to the concentration. You'll never reach negative concentrations with decay, never. Only with sink. And if you're modeling glucose concentration, you don't want your glucose concentration to go negative for obvious reasons. All right, demonstration. One cell absorbing and the other cell secreting. So our objective is to build this simulation over here, where one cell secretes, the other cell absorbs, and we can, for example, plot, what am I plotting here? I'm plotting the amount of field up, the amount of material taken up by the uptaking cell. Okay, so it's a demonstration, not a guided exercise. So I will demonstrate for you. If we have time left, please go ahead and download the simulation run by yourself, or we can do a guided exercise where I do the step-by-step with you. So, okay, I will start over a new simulation, close this to edit. CC3D project, new CC3D project. Pedro uptakes energy of students. Let's reduce the lattice because we want the simulation to run faster. The boundary conditions here doesn't matter because they have no effect for the field. Rectangular slab. Let's set here cell A and cell B. Next, a chemical field. Let's put here VEGF. Let's set here, we don't need contact and volume, but we set it nonetheless. And secretion. Secretion you must have. Even if you're only modeling uptake, you have to import the secretion steppables. Okay, so we don't need to set anything here because we'll use the Python. All right. So this is my simulation. Close here. So again, a bunch of boring stuff. For example, I want to freeze the cells, so I can type freeze equals freeze equals quotes. Now, okay, nothing secretion, that's good. In the VEGF, let's put a high global diffusion because I want to see things happening fast. Global decay 0, I don't want decay. I already have a cell taken up for me, so I will leave it as 0, so I can even comment it, comment this, comment this, comment. I don't need this as well. Let's remove everything. Secretion here, not needed. I can even delete everything. So look, it's much nicer here. And just to vary things, I will set constant derivative 0. So nothing goes in, nothing goes out. We didn't use this so far, so I will use the constant derivative 0 because we vary a little bit more, okay? So we must create 2 regions of cells, as I'm showing in the video. So 2 cells, one the left, one the right, two little squares. To do this, I have to set 2 regions, 2 separate regions. Cell A, I can define, for example, from 20 to 30, 40, 45 to be symmetric to 55, a width of 10, and only one of the cells, cell A. And in the opposite size of the lattice, or from 70 to 80, Y from 45 to 55, only cell B, width 10. Can you relate to the image? So 1 little square on the left, 1 little square on the right, right? 10 by 10, each 10 by 10, okay? All right. Here in the steppables, we only need to make 1 cell secrete, the other cell uptake. So let's go and take the syntax. So secrete inside cell. We make 1 cell secrete. Again, the secretor object, put it outside the for. You don't need to get the secretor object every time. So here you set the name of the field. In our case, it's VEGF. Let's just make sure it is. Okay, VEGF. And here, let's set only cell A to secrete. So I have to change this. I have to filter the cell out. Instead of looping over all cell list, I can do cell list by type. Or better than that, instead of writing 2 loops, I can do if cell type equals self.cell A. So if my cell is of type cell A, it secretes at a rate of 0.1, right? If cell type is self cell B, instead of secreting, I want it to uptake. So let's get the syntax: uptake inside cell. Yeah, uptake inside cell. I already loaded the secretor plugin, the secretor object. I already loaded the secretor object with name VEGF. Okay, so I don't need to load again. I can delete this line. Right? And here we have three arguments. The cell we want to uptake, the saturation sync rate, so it's the cell object, comma, saturation uptake, and the relative uptake, right? So the relative uptake takes place if your field is low, and the saturation uptake takes place if your field is high. So in other words, your cell can only take up an amount of two of that material per pixel per Monte Carlo step. Okay, so I'll even write it down here. So the saturation rate, maximum absorption for each pixel, each Monte Carlo step, and the relative rate, basically a decay. So if you are under this amount, under this threshold, your cell will uptake like a decay, right? So let's run the simulation, see what we got. How much time we have left?
You have 30 minutes.
Sorry?
You have 30 minutes.
We have plenty. Nice.
Yeah, we end at 6.
Yeah, so let's take a look of our field. I may even put more isolines because I find them beautiful. Beautiful. So fields, isolines instead of 3, let's put 10 isolines. Let's put 20 isolines. Good. And instead of fixing our field between 0 and 1, I rather free the field concentration to see where it goes. So tools, configuration, and unfix both. So as you can see, one thing strikes my eyes, which is the minimum concentration. Our minimum concentration is not 0. Our minimum concentration is 0.2. Why is that? So Shirley asks how I add contour lines. So basically tools, configuration, field tab, then scalar contours, click show, and set the amount of isolines or the value of the isolines you want to see. So tools, configuration, field, scalar contours, show, set. You still didn't answer my question. Why is the minimum concentration 0.3 if the cell is taken up at a rate of 0.1, 0.2 per second per Monte Carlo step? Why is it not zero inside the cell? Yes, relative value set. That's the proper answer. It can go to zero because of max uptake rate. That's not the reason. Actually, we are far from our maximum uptake rate still. We are far from it. I have to set a very high secretion in the left cell so we can see the blue cell, the right cell not being able to take up all this amount of field. So for example, so let's take advantage of Sharon wrong answer and set, for example, an uptake, a maximum uptake of 1, but our left cell secretes 100 per pixel per Monte Carlo step, okay? Per pixel per Monte Carlo step. This is important. Now let's run and see what Sharon was saying. So if the amount of field is so high that the right cell cannot absorb, we will see this number rising, 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 and will never stabilize. In the other case, it did stabilize. Remember, Sharon? In the other case, that number stabilized around 0.3. Now it grows, 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 grows because the cell cannot take up everything. So 400, here's 1,000, 500, growing, growing, growing. And we can see that it reached steady state. We don't see these isolines moving. Do you agree, Sharon? We reached the steady state here, but the field keeps growing in intensity. It was in the cell because we were looking at the minimum field value, which must occur inside the cell that is taking up the nutrients. So I will get back to the previous example. So a maximum uptake rate of 2 here and 0.2. Let's equilibrate things here. So now the cell on the right can take up everything that the cell on the left produces. So in this particular case, the minimum value, the value of field, the minimum field value must stabilize when we reach the steady state. So it will stabilize. Now it will not be 0.3 because I set a different secretion rate for the left cell. It will be probably double that, 0.4 something or even higher. I am wrong. Yeah. Look, it's stabilizing. It's not growing as faster as it was. Okay, so because the left cell and the right cell can equilibrate because the amount of material produced by the left cell can be taken up by the right cell. The system equilibrates and you reach both an equilibrium for the minimum and maximum values, okay, Sharon? But even in this case, we don't see a zero concentration in the right cell. That's because the field is diffusing inside the right cell, and the right cell doesn't have a high enough decay constant to make the field go to zero inside. To make it happen, we could, for example, increase the relative uptake. Let's say 1. So this may be enough for the right cell to take up so fast, so quickly, that inside the cell, we always see zero concentration. So in this example here, we can see that our minimum value is still being held at 0. That's because the right cell uptakes at a too high pace, right? Is it clear now? I'm not sure about the answer to this question, Sharon, but maybe could be the case. I'm not sure. I'm not sure. No decay inside the cell is like no consumption then? Perfect. Yeah, that's right, Shilei. Shilai, first or second? I might not be on the right page. I will watch the video and think about it more. Let's think. Second one, Shilai. Okay, Shilai. Perfect. I prefer Shilai. Shilai is too American. Schlei. So we still have time, I think almost 20 minutes left. So you can take advantage and run the simulation if you want, downloading from the student materials. Or if you want me to test something, more cells, I can make some tests here if you're curious. Make questions, unmute, everything. You can do everything. Free time. Okay, let's build a simulation then. So this is a good suggestion from Professor Glazer. Let's build a simulation. Step by step, like we are used to do. Okay, so first step, import the secretion plugin. I will create a new simulation here, new CC3D project. All tired but Pedro. Can leave lattice dimensions to 56, no problem. Everything okay here. So next, set cell A and cell B. Definitely, Ghostmate, definitely. I will freeze here. Okay, understandable. So I will get back here. Don't change anything here in the general simulation properties. I am restarting the simulation, okay? So don't change anything here. Hit next, set cell A and cell B. I'm sorry, Ghostmate. Yes, unfortunately, it's hard. Everyone should go to the same place if we wanted to do the workshop at the same time for everybody. The problem is that we would have less students, and it would be much more expensive, right? So. If James pays, I agree. I vote yes. Okay, next step. Chemical fields. Set a chemical field, VEGF, I don't care, anything. Yes, it is this exactly the same I just did. Schlein. So let me make sure you guys are on the same page as me. All right, let's move on. Select contact, volume, flux, and secretion. Hit next. Don't do anything in the secretion plugin. Next, finish. Open your simulation and go to your XML. Module poll premature. I was trying to answer this. I don't know, Sharon. I don't know. Maybe I did something wrong. I'm not sure. Okay, so if you're unable to follow, please type in the chat where you are stuck, and we fix the problem right away. Okay. I'll move on. So if you have problems, please type in the chat and we solve the problem. Okay, so here, adrenaline secretion rate too low. Okay, you can set a very high lambda volume for cells. So for example, 100, or you can freeze the cells. Either one works. 100 is fine for lambda volume. So again, set high lambda volume so cells stay still. They don't move. Now go to your diffusion solver FE and delete everything that is not diffusion constant, diffusion global, and the boundaries. Get rid of everything except of the boundaries and the global diffusion. So you get rid of global decay, you get rid of diffusion for cell, you get rid of decay for cell, you get rid of secretion, you get rid, yeah. And in the boundary conditions, set constant derivative 0 for all boundaries. So constant derivative, both in X and in Y, 0. All of them, 0. So the boundaries act like walls, okay? I'll give you a minute to accomplish these tasks. So comment everything that is not related to diffusion, global diffusion, and the boundaries, and set constant derivative to 0 in all boundaries. Yes, Tamaya, you get the wrong one. So please copy the syntax here and paste on the other one. You got this constant value, like this constant value. So what you have to do, just copy this constant derivative, double click, copy and paste here. Or you can simply copy these two lines and paste in the two lines above, over the two lines above. Okay, so far? Let me relaunch the poll. Why did we get constant value? No, you did nothing wrong. So this is the default. This is the default input from to edit. There's nothing wrong. So absorption, absorption with a B. So I will even give you another one. Adsorption. This is another one. Adsorption. So you have 4 concepts. Absorption is when something outside goes inside something else. This is absorption. Uptake is like a cell eating something. It absorbs, but it also destroys. Okay? It absorbs and destroys. Transport is happening even if you don't have absorption, even if you don't have uptake. Transport is like if something is diffusing, it's being transported. Okay? And what's the difference between absorption and adsorb? It's difficult to write these words. Absorb. Each letter in one side of the keyboard. It's very strange. Then make sure you guys are on the same page as me. So adsorption is when something sticks to the surface. This is adsorption. It stays outside, but it's on the surface. Absorption with a B, it's inside. Yes, that's it. Perfect, guys. Let's continue then. Yes, Hayden got a point, so yes. Now here in the region, we must create 2 different regions, 1 cell in the left, 1 cell in the right. So what you need to do is set, for example, X from 20 to 30 in one, from 70 to 80 in the other, and Y, let's be symmetrical, 45 to 55, 45 to 55. And in the first one, cell A, in the last one, in the right one, cell B. I'll give you a minute to, another detail with 10, actually it doesn't matter. Anyway, with 10. No, in this case, it does matter, yeah. Okay. I'll wait a minute for you to complete the uniform initializer, so we can move on. If you're not ready, please vote you're not ready. This is important. Otherwise, in 3, 2, 1, okay, let's go ahead. Steppables, get rid of the print. Print is useless in this case. Too many zoom little windows in my screen. This is irritating, but okay. CC3D Python, get two syntaxes. Secrete inside cell. Secrete inside cell. And the uptake inside cell. Get rid of the nonsense. Yes, we are in the steppables.py, which is a Python script. Yes. Import the two. So yet again, as I was going too fast, go to CC3D Python, secretion and uptake. And here we have two things we want to import. First one, secrete inside cell. The second one, uptake inside cell. Import both of these syntaxes, okay? I will leave the mouse hovering for a second. Okay, once you import these lines, notice that we only need one secretor object. To where? So inside the for loop in the step function. As we only need one secretor object, you can delete one of them and pass the other secretor to outside the for and give it a name. In my case, it's VEGF. Let me just make sure it is. Yes, it is. VEGF, get the secretor object. Okay, that's it. And now we can set the secretion constants, 0.2, for example. So let me make sure you guys are on the same page as me. Yes, yes, indeed. I will delete everything here and start again. So CC3D Python. So look here. I am in the step function inside the loop for cell in self-cell list. Okay, I am inside this for loop. Then I go to CC3D Python, secretion uptake, and import the secrete inside cell. Click here and delete the nonsense. Like we have these comments here, delete. And now we are with two lines, okay? Now, over again, CC3D Python, secretion uptake, and now uptake inside cell. delete the nonsense. And there we go. So we only need to get the secretor object one time, so you can pass the secretor outside the for, outside the loop. And give it the proper name, because our name is called VEGF, or whatever you called. So you could have called him Pedro Handsome. So I wouldn't argue against that. So in this case, you will type Pedro Handsome here as well. Once you take this object, these three functions are working. Now set meaningful secreting rate here, 0.2. But notice that all cells are both secreting and taking up. Both cells are secreting and taking up. It doesn't make sense. We want the cell A to secrete and the cell B to take up. So if cell type is cell A, secrete, so we have to tab here. So the secretor is inside the test. And if cell type is cell B, it uptakes. Shlai, no, we don't need to do this. We can do this. Self.secretor, pass it to the start function, and self here, self here. Then you don't need to call it every Monte Carlo step. Yes, this is one extra layer of complexity that I didn't want to do, but this one is a little bit easier in terms of coding to understand. But yes, it's a bit less optimized. So Shlai, please do this in your code. If you want, okay? So let me make sure you guys are on the same page as me. Yes, it's not that slow, so doesn't matter, right? Let's make sure you guys are on the same page. So if you have everything set, which is the case for the majority of you, left click, open in player and run. And it didn't. Oh, okay, so I have to, yes, run new simulation here. Oh, crashed. Yes, sometimes it will crash because, you know, programming without crashes doesn't make sense. is not programming. Let's open again. Secretor is not, yes, that's right. Yes, now that I am inside the function and not outside the function, I can define the secretor, and this was a bug. Oh, oh yeah, I didn't set my lattice to 100 by 100. I think it's a problem for you too. So in order to run like it was in the video, that's my mistake. Set 100 by 100 in the XML. Now it will make sense. Yeah, there we go. So this is our system. And if you want to check out the VEGF field, it's very slow, very slow. So in order to make things faster, you can go ahead and increase diffusion 100 times. So put a 10 global diffusion constant, 10 global diffusion constant. Then you will see it happening faster, hopefully. So I made 2 quick changes. I changed the lattice to 100 by 100. And I set global diffusion equal 10. That's all I did. And it would be interesting to plot a graph in the X coordinate. So we will see something like oh, really? It takes so long to open Paint? Do you even have Paint? Ah, here. Ah, the simulation is running and the notebook is not so great. So yeah, here. So the graph would be something like this. So this is X coordinate. This is Y coordinate. So the graph would be something like this. If you plot the graph in the X dimension. You have, yes, all capital, yes. Could you guys reach this result? I will show my code then here in the steppables. Here is the if. Yes, sure. And then after that, I changed 2 more things. I sent to James directly, so after I implemented the if statements for secretion and uptake, I also set 100 by 100 lattice and global diffusion to 10. Uh, Go to your XML, Edgar, and check if you set the proper name for the cell. So, for example, here I have this name, cell A, here in the cell type, cell A and cell B. Check if you spell that correctly. It must have the same spelling, but in the Python, everything uppercase. Check if you are using the double equal or the single equal. Makes a huge difference. It must be all uppercase, Edgar. So self dot cell A like this, self dot cell B like this. Or you can type 1 and 2 if you like. It works the same. If I put here 1 and 2, the simulation works. I can run it normally. Works fine. Both work. You can type 1 and 2, but the problem, of course, is that you don't know. Sometimes you don't know which cell is the type 1, which cell is the type 2. So you can do this. And I saw one guy doing this. For example, you could do cell A, or for example, left equals 1, right equals 2. And then instead of typing this, you could type left here and right here, which is easier to remember, okay? So that's it for today. If you have any question left, please type in the chat. Otherwise, please fill out the questionnaire. Again, it's very important for us. Yeah, go ahead, Goldsmith. Yes. Yes, you can unmute. Yeah, no problem. The difference between defining in the XML and Python is basically flexibility. Okay, that's the difference. So in Python, you can do a bunch of other stuff. So for example, you can secrete if the cell, for example, proportional to cell size. No, this is already made, but for example, the cell secretes only if it has another field inside, let's say. So in this case, the Python is more powerful.
Or you could have the rate of uptake or the saturation level be different for each cell.
Or
Or even,
time, yes.
or even the rules could be different. Some cells could uptake everywhere, some could uptake only on contact. Now, something that's a bit different that I put in the chat that's maybe worth repeating is that for secretion and uptake, if you define it in the Python and in the XML secretor and in the diffusion steppable, all of them will act. All three of them will act. And so they don't replace each other. They're in addition to each other. Sometimes that's helpful. Sometimes it's not important. Occasionally it's confusing. But in fact, there are three independent secretion solvers. And so they're all possible to turn on or not at once. I have a slide, and I don't remember off the top of my head what order they're executed in. But there is a specific order in which they're executed. So if you turn on secretion in two places, it will secrete twice as much.
I have a question about the ways of implementing something like cytokine signaling, where you can't use uptake because the let's say the signal being interferon, it's not going inside, but it's interacting with its receptor, and then that activates downstream stuff. So how would that be implemented, assuming that each cell has let's say an SBML model inside it?
So you have to decide, the key question is the ligand destroyed or not when it's received? If the ligand binds and then is released, you measure the concentration at the surface of the cell. So you multiply that concentration by the concentration of receptors, and you calculate the rate at which the signal is sent. That would be non-destructive. If it's destructive, you would have an uptake rate at this outside the cell at the surface that's proportional to the concentration of receptors, and you'd have it return the total amount.
Yeah. So does like the method called amount seen by cell, does it measure the concentration inside the pixels technically inside the cell, or does it measure at the surface, because the signal would interact with the receptors at the surface of the cell, right?
You have a choice. You can have all of the different permutations.
Okay.
If there's a permutation you need that's not available, then my suggestion is to have it uptake some tiny fraction of what's there. So it's an negligible perturbation. Have it take up 1/1,000,000th of the amount at the surface, which doesn't really change the amount at all, then multiply by 1,000,000 and you have the amount at the surface. So if there's some particular configuration which doesn't let you do it non-destructively, just take up a very small fraction of what's there, and then you scale it.
Okay.
And that works fine.
Okay.
If you take up a million of the amount that's there, it doesn't perturb it significantly.
Yeah. Okay.
Most of the possibilities are implemented both ways, but not all of them. And so if you have one that you really need that's not implemented, you have to come up with a little bit of a game to play with it.
Yeah. Okay. Yeah. Cool. Yeah.
But that's a great question because, of course, if you have an extracellular receptor, and it's of course true for cytokines, but it's true for many growth factors and other molecular species, you have a transmembrane receptor protein that binds to the ligand on the outside of the cell. And so then that's specifically why we have uptake at the surface outside the cell as an option.
Okay.
Great question.
So another slight question adding on to this, the method called amount seen by cell in the secretor plugin, what exactly does a cell see mathematically when using that method?
Well, so it is not rescaled automatically by the number of voxels. So if you want to know what the density seen is, you have to divide by the surface area of the cell or the total area of the cell. Because it will be literally the sum over each pixel, each voxel of the concentration for whatever domain of concentration you define. The whole inside of the cell at the center of mass, at the surface outside, at the surface inside.
Okay.
And so as the cell changes size, for the same concentration, as the cell changes size, that will return a different number.
Okay.
Now if what you want is something like a signal density, that's not what you want. If you want to know what's the total amount of glucose I've got available to me, then that is what you want.
Okay.
So one thing that happens sometimes with CompuCell that makes things a little bit more tricky, perhaps, is that people use it in all sorts of different ways. So we try to make it very flexible, but sometimes that flexibility means that you have to make a lot of decisions about what to do, or the plugins get complicated, like all the permutations of absorb inside, absorb outside, secrete outside, secrete on contact with, and so on.
I'm asking this question because if you remember, we met once as a part of a meeting with Dr. Shoemaker. I worked with him. So, yeah.
Right. I think it's a great question, and I think I'm happy to work with you separately about this too. But I think it's a topic that will be of interest to many people who are taking the class. So I think that's great to ask.
Yep. Thank you for your time.
I'm working on the problem too. I hope you can talk to Giuliano a bit about it because he's of course worked on these issues as well.
Yeah, I'll definitely talk to him too. Yeah.